One of the most important steps in cancer research is interpreting the results of research studies. This makes the Comparative Pathology Shared Resource an important partner in the search for new ways to prevent and treat cancer.
Pathology is the study and diagnosis of disease through the examination of organs, tissues, fluids, and whole bodies. So comparative pathology is the study of disease in animals, particularly as those diseases relate to the human conditions.
What makes the Comparative Pathology Shared Resource particularly valuable is that its services extend far beyond evaluating tissues from animal models.
So we offer expertise in the planning stages of the experiment, even in grant writing, and all the way through to the publication of results and assistance with the manuscript. All of our lab technicians are highly trained and very experienced. Our current pathologists all have PhD degrees and are boarded in veterinary anatomic pathology, and most of us have been PIs on NIH grants, so we understand the challenges of NIH-funded research.
The way the Comparative Pathology Shared Resource helps investigators with their projects depends on the goals of the project and the individual investigator. Most commonly, it's the evaluation under the microscope of tissue sections to provide a diagnosis of the tumor type, its cell of origin, and its behavior, which organs it's affecting, and whether it's extending into other tissues.
First, the investigator should contact one of our pathologists to explain the goals of the project and to ensure that there is a plan to correctly collect appropriate tissues. Then, once the study is completed and the tissues are ready for analysis, either we would help with the necropsy exam and collect the tissues, or they would be sent to our laboratory. Paperwork would be filled out to indicate which tissues are being sent, animal identification, etc., and whether or not pathology interpretation is requested.
Once the fixed tissue samples are in the lab, they're trimmed into small pieces and embedded in wax. Cutting equipment is then used to slice very thin sections. These sections are placed on slides and then stained in one of two ways, by hand or with an autoimmunostainer for projects with many samples. The lab can also process frozen samples.
Once the sections are prepared, they're sent to the pathologist for evaluation, and then we communicate with investigators either electronically or by telephone. We also have a pathologist who once a week is on site at the Masonic Cancer Center. This occurs Tuesday mornings, and she's available for drop-in consultations.
Masonic Cancer Center researcher Jaime Modiano is currently working on 3 projects with the Comparative Pathology Shared Resource.
I think it's very important for us to collaborate with a Comparative Pathology Shared Resource for 3 reasons. The first is that they provide professional expertise that is not widely available elsewhere, and so we don't need to duplicate that expertise. The second is that they provide technical expertise, and so we don't have to develop all of these assays in our lab. And the third, which is equally important, is that as we discuss these projects with them, they can have ideas that we may not have thought about that can take our research in new directions and into new discoveries that would be impossible if we didn't have these type of collaborations.
With the right facilities, equipment, and people, the Comparative Pathology Shared Resource is enhancing research at the Masonic Cancer Center and helping to make a difference in the lives of cancer patients.